Screen. Hello, everyone. Can you see my screen?
Yep.
Okay, great. Hello, everyone. Thank you for joining the IUPAC WorldFAIR Chemistry virtual webinar series. My name is Fatima Mustafa, and I'll be hosting this event. This is actually the fourth webinar in our series with the title of What is a Chemical? And this time, the title of our webinar is Innovation in Chemical Description. And as you can see here, we are joined by a group of experts and a group of amazing speakers who will be introduced separately by our moderator, Ian Bruno, and I would take this opportunity to acknowledge them all for kindly accepting our invitation to be here with us and enlighten us about the new technologies and innovations in the chemical descriptions. Thank you all for being with us today. So we will be starting by flash talks by each of our speakers for 5 to 6 minutes. Those will be followed by a panel discussion. So please feel free to type in your questions in the chat, and also you could also share your notes with us. The link is already provided in the chat. Those notes also could be provided after the discussion, and those will be available in our Zenodo community as well. We will be close the meeting with having your feedback and giving you some announcement about our future activities. So for those who have not been with us before, this project is led by the IUPAC. And the IUPAC is the International Union of Pure and Applied Chemistry. The IUPAC is the worldwide authority of chemistry, which main role is to take care of all of the chemical representation, nomenclature, terminology, and critically evaluated data. Since over a decade now, the IUPAC is trying to translate all of those standards into the digital domain. So with that effort, one of the projects that have been involved in is the WorldFAIR, which is a kind of new project, a global initiative led and coordinated by the CODATA and the REA. And the main role of this project is to advance implementation of FAIR data principles. I would take this opportunity again to thank all of our volunteers and experts from all of the collaborators you see here who are working with us toward these goals. So our project, the WorldFAIR Chemistry, main goal is to make IUPAC asset fair. We have 3 subcommittees or sub groups are working towards reporting. One of them is called the reporting guidance, in which they are developing guidance protocols for handling chemical data fairly. The second project is the training cookbook, which they actually develop online materials and you could say recipes on how to manage digital data files and content. And the third project is the protocol services, in which they are trying to develop web-based services to confirm chemical identity and machine readability for chemical data. You can actually read and have an overview about our project entirely in the latest issue of the Chemistry International. As you can see here, there is the link. You can enjoy the reading. So from here, I would like to introduce our superstar moderator who have been working to moderate these webinars in this series, Ian Bruno. Ian, by the way, is a lead of one of these projects, subcommittees in the WorldFAIR Chemistry, the guidance protocol. And he is, of course, in the field. He got his bachelor degree from Durham University and PhD when that time was called Information Science from University of Sheffield as well. He has been worked in Cambridge Crystallography Data Center, the CCDC, since 1993. And his current role right now is the Director of Data Initiative, where he is responsible for establishing strategies for making CCDC data more readily discoverable and reusable. He is actually involved in many organizations and many unions, such as the RDA, the International Union of Crystallography, and the IUPAC. He's currently the Secretary of INCHI Trust. So Ian, now the floor is yours.
Thank you very much, Fatima, and I would also like to thank you for all the work that you've been putting in to bring the speakers together and set up this particular webinar, and indeed all the other ones that have come before us. Before we get into the speakers, we just wanted to do a little poll, just to explore some particular areas to have an understanding of how important they are to you in your research. So there's 2 questions in this poll. We're asking what's important to you day to day in terms of representing complex systems, classifying molecular entities, and mapping chemical concepts and data types. And if these are important, or generally how familiar you are with technologies associated with these concepts. So if none of those are relevant to you, you can probably say at least answer the second question, if not the first. And while we have that poll filled out, I think with some of these talks, we're going to go a bit away from what we've perhaps focused on in some of the earlier webinar series, where I think and indeed a lot of conversations about representing chemicals, which is to think of discrete, well-described organic chemical structures, and start thinking more about the more complex systems that people encounter in their day-to-day research, which is the reason for the poll questions here. How many people have responded so far? Cool. So if we have a look at the results that have come in so far, 60 to 70% of you, these kind of concepts are important. And then we look at the familiarity with the technologies that are out there. Maybe, it's not particularly high. So I think truly we are going to be hearing about elements of representation of chemicals that are perhaps more on the innovative side. So thank you for your contribution to those poll questions. I will now move on to start to introduce our first speaker. And I think when it comes to innovation in the area of chemical description, Henry Rizapa is someone that I would put at the forefront of someone I would choose to talk about this, because he himself has been at the forefront of opportunities offered by the digital age. He was an early adopter of computational chemistry, a pioneer in semantic representation of chemical data, and has been a strong advocate for the importance of good data management practices. One of the key parts of this has been understanding what it means to chemically enable the internet. Part of that involves a concept called media types, and that is what he is going to talk to us about today. So Henry, over to you.
Here we go. Well, I'm doing it slightly differently. I'm probably the only person who isn't using a PowerPoint or PowerPoint-like tool today. I thought I'd share it to you in an interoperable, reusable, accessible, and findable way by simply assigning it to DOI. So if anything grabs you and I've already moved on in my slides, please make a note of that DOI JVK9 and you'll be able to access what I'm talking about. So I'm going to start with what is a media type. It's been renamed since the original days, but it goes back to the year dot. In fact, although the final document is dated 96, it started a few years before then. And it's a way of trying to map an association between a document file name and what they called a media or MIME type. And it was a hierarchical system. It had a primary type and a secondary type. Since the original days, a suffix has been added to the subtype, and then also parameters, although these are very rarely used. So that's what a media type is. And to make it more concrete, I'll give you an example of how it could be used even today. So this is a command line which has a media type in magenta, and then a target server, which happens to be a DOI. And you can basically invoke that command line to get, in this case, metadata. To summarize, it provides actionable mappings between metadata as specified by a persistent identifier and wait a minute, and a file content. So part of my screen looks good. So in retrospect, if you like, it's a perfect sort of synonym for FAIR in some ways. By putting in metadata, which consists of media types, you can find it according to the media type by searching metadata. You can access it by requesting that type of metadata called content negotiation. You can interoperate it because you've got contextually assigned file formats and reuse it, which is sort of the same thing. But there are issues. And I'll show you what the issues are. And thanks also to Ian for pointing out this resource at the top. You type in an extension, which is CIF, the crystallographic format, and you're immediately told by this particular resource that there are two well-used definitions of CIF, completely orthogonal. Neither will interoperate with the other. One is essentially a CD creator image, and the other is the crystallographic information file. And there are numerous other examples across numerous domains which use the same suffix, but where the content is essentially orthogonal, and they need disambiguation. Also, many document types often have multiple suffixes. Famous ones are JPEG and JPEG, TIFF spelt with 1 and 2 Fs, etc. So there are ambiguities there which make it difficult for machines, especially, to cope. So way back in '94, we proposed that the properties of the chemical media type were so unique and so specific that it was useful to have that as a primary type. Quite a bold proposal in those days, and a typical example proposed then was chemical/CIF. The IETF wouldn't let us use that because it was non-standard then, and so the non-standard was designated by the X. The media type's configured server-side based on the file name extension, but also based on the contextual content. In other words, the server is presumed to have other information which tells it the likely content of these file name extensions. In our case, we put that context in using ELNs and databases. And it achieves a level of abstraction between the file name suffix and interoperable content that can now be declared as metadata. This is how it works in practice. There's an API there, that's the top link, is the API where you work out how to do it with DataCite, that is. And I'm going to show you some examples, and you can try these out live. So this is the total number of media types registered with DataCite, and it's approaching 1 million. So that's gives you some idea of the uptake. If you do a search for chemical media types, you get in excess of 200,000, which is actually quite sort of a good amount of data. And I'm going to show you how it can be combined. So you've got these Boolean operators, and here you've got a number of media types which in the context are assigned or are thought to have NMR content. The JCamp is well known, it's an IUPAC standard. This is a commercial sort of MNOVA, various things. And even there, you've got 978 works. So this is how media types work. So moving on, these are examples of some searches. And the reason I put them there is you can combine these media type searches with other properties. So the first three are 3 different ORCIDs, the researcher. Here you can combine 3 media types associated with calculations, Gaussian checkpoint, Gaussian log, and Gaussian input files. So you can find 6,648 that have all 3 of these media types present. And here finally is a combination of NMR data and Gaussian. So you can search for media types containing Gaussian and media types that may contain NMR data. So that's how you find it. That's the F of find, how media types can be used to find things. Next to the access, this comes in two levels. One is the human level, where the human is directed to a landing page for some data. And here the media type is simply listed, and the human can select however many of these they wish to have. So that's sort of the human aspect of media types. But there's also always a machine aspect, and it's buried deep in the metadata. In our case, it comes with two types for technical reasons that I can go in the discussion, but I won't go there now. The first is to declare that any particular metadata record has additional metadata or related metadata. And that's an ORE record, and the ORE record contains the media type declaration. So that's how a machine could use this as well as a human. Next we have interoperating. And I want to show you this, and it's actually a triple slide. So this is a table that we prepared for a very recent publication. It contains the typical sort of columns and entries that you find in journals, but they're all active. So let's presume that you go to this thing and click on a number, and it expands to give you a toolkit. It expands to give you not only a toolkit, but a molecule. So here we have the molecule. Open by clicking on that. Here is the toolkit. Using the toolkit, you can save the file or do lots of different things with it. And my final click will be to go to the actual live version of what I've just shown you. It has its own DOI, has its own metadata record. Here is the same thing, but now it's an active thing. Oh Lord. Now, the best laid plans can go wrong. How many minutes have I got left? My mouse, which is battery operated, has just run out of charge. I hope that 20 seconds worth of charge will give me enough charge to go right in. I knew I should have charged the thing before I started. Most people run out of charge in their laptop. I'm running out of charge in my, here we go. Right, click on that. It's retrieving the data using media types. And I'll show you how that works in a minute. And now you have the thing, and you can open the toolkit and so on and do lots of lovely things. I'll just show you the actual code that's needed. So into your web page, you simply use a script and the DOI of the data, and it then works on media types. It goes away and gets the media type and loads it into the program and interoperates it. So that's more or less an indication of how media types can be used out there to sort of enhance all four aspects of FAIR. So it can analyze the findability using searches, the accessibility using various metadata records, the interoperability, you can load it into software, and ultimately, as you can see here, media types can also be considered as a useful node in data-oriented knowledge graphs. So that's for the future. And the last link shows you summarizes some of the key papers that we've been talking about over the last few years. Okay, that's me done. Thank you very much.
Thank you, Henry. And so we're going to keep moving on with the presentation. So if you do have specific questions for Henry, please feel to put them into the chat or the Q&A, and we'll make sure they get addressed as we go along. But I think that was sort of, a great example of how established frameworks put together to run the internet generally can, once you, with a little bit of sort of like digging, be used to provide sort of domain-specific solutions that address particular needs, for example, in this case, chemistry. So thank you very much for that. Our next speaker is Adnan Malik, who works at MLB EBI. He's got a background in interactions between big biological molecules, and he's worked as a research scientist in academia and in industry, where his focus was on anti-malarial drugs. At Emble EBI, he's the curator of the KEBI database, and I would argue perhaps that KEBI has been one of the innovators when it comes to ontology-driven classification of chemical entities. So looking forward, Adnan, to hearing about KEBI and where it is today. So over to you.
Okay. Can you see my slides?
Can indeed, yes.
Okay, that's good. So I would like to thank Fatima and the organizing committee for inviting me to this webinar. So I'm Adnan. I work at the European Bioinformatics Institute in Cambridgeshire. So in this talk, I'm going to be talking about KEBI. KEBI is a database and also an ontology of chemical entities of biological interest. So the chemical entities in KEBI are either natural products or synthetic compounds, which may be of relevance to the biology community. So we have two major tools in KEBI. We have a submissions tool, which enables users to submit new chemical entities to the database. And we have an internal creator tool which we use to annotate those entries in the database. And then we also have the KEBI website where users can access the information. So when a new chemical entity gets deposited into KEBI, we give the entry a 2-star status. What this means is that the data that's been provided has come from the submitter. So once the entry gets manually created and checked by a member of the KEBI team, we then assign the entry as a 3-star entry. So this means that the entry has now been manually checked and verified. So what does KEBI provide? So this is just an example. So this is nicotine, an alkaloid found in tobacco. So we'll provide a 2-dimensional structure of nicotine. We'll make sure the stereochemistry and the structure is correct. We'll also assign an unambiguous KEBI name to the entry, and we'll also provide a unique and stable KEBI identifier. If a crystal structure is available, we can also add a 3-dimensional structure of the molecule. In addition to this, in terms of chemical nomenclature, obviously there's going to be several different names of this molecule. So we'll provide a recommended IUPAC name for the structure. For pharmaceutical drugs, we can also provide international non-proprietary names in several different languages. And then we'll also extract synonyms from the primary literature or from other resources. For example, these can be either non-systematic names, common names of compounds, and also research codes. And then we can also provide brand names and trade names of molecules. In addition to this, we'll also provide some structural properties such as formula, charge, mass, and also some identifiers such as INCHI, INCHI-key, and SMARS. For natural products, especially the ones that have been detected or isolated from living organisms, if we can find data on this, we'll also provide the species where this molecule has been detected in, and also provide a link to an appropriate taxonomy for that species, and also provide a link to an appropriate citation where we obtain this data from. KEBI also provides database cross-reference links to around 65 different resources, and some of these resources are domain-specific. For example, if the entity is found in those resources, we'll map to that resource from KEBI. So a lot of these mappings are done by Unichem, so we map via INCHI-keys. And we also provide registry numbers to some commercial databases, such as the Chemical Abstract Service and registry databases. We also provide some relevant citations, so any publications which mention the entity, chemical entity, are provided with a link to the full article via EuroPMC. So this is a text mining tool we developed several years ago in collaboration with EuroPMC. So what this tool does is it highlights the chemical synonyms of a given chemical structure and identifies articles, identify those synonyms in the titles and abstracts of research articles. And then a KEBI creator can then go through the list and add the relevant articles to the entry. So in addition to this, any chemical structure which comes into the database, we also assign it within the KEBI ontology. So the KEBI ontology is a chemical ontology where we classify molecules based on the molecular structure and also the biological function of that compound. So here is just an example of osimertinib, a drug used for the treatment of non-small cell lung cancer. So the structure of osimertinib is shown on the left-hand side. So what we do, we actually break the molecule down into its various components and functional groups. So if you look at the green circle, so that part of that structure is an indole. So we'll put this molecule into a class called indoles. The red circle is an aminopyrimidine, so we put this molecule into a class called aminopyrimidines, and we can repeat this process until all the functional groups are covered. So you have more specific classes at the bottom, and then you have the general classes at the top. And we can do the same thing with the roles. So each molecule, if we come across its role in the primary literature, we'll then add the roles within the ontology itself. So osimertinib has a role as an EGFR antagonist and also has a role as an anti-neoplastic agent. So here I've just shown you a very simple view of the ontology for a given compound. I'm only showing you just the two relationship types, but in TIB we have several chemistry-specific relationships to define relationships between, for example, protonated, deprotonated structures, as well as tautomers and also enantiomers. So the ontology can be, so the ontology is actually more complex than this. So when you have, for example, thousands of molecules being classified within the ontology, you can carry out some very high level queries. For example, one can search for example, all indoles in the database which have a role as an anti-neoplastic agent. This ontology is actually manually assigned by a chemistry curator, so it is a very tedious and a slow process. So finally, I would like to thank MBLEBI and BBSRC for funding, and all of these, acknowledge all of these individuals who have played a vital role in TIB's maintenance and development over the years. And thank you for listening.
Thank you very much, Adnan. And I think that sort of highlights how, some of your comments towards the end there, how the importance of thinking not just about a chemical as a structure with those structural components, but also its wider context, and then the opportunity that there is to bring kind of technology to sort of use those to make inferences that might otherwise be difficult to make. So thank you. If you have specific questions for Adnan, drop them in the chat or the Q&A, because we'll move on now to hear from Oliver Koppler, who's going to talk to us some more about ontologies and the opportunities that they present. Oliver's career has taken him through the development of software, databases and infrastructure for chemistry and chemical information. His current focus is on knowledge engineering through lab-linked scientific knowledge at TIB University Library. As part of this, he's at the forefront of the NFDI for Chem project, and he's going to talk to us about ontologies for Chem. So Oliver, over to you.
Thank you very much. So hopefully you can see my screen.
Yes, can.
Perfect. So yeah, thank you very much for the opportunity to present the work we do with ontologies for Chem and NFDI for Chem. So our work is motivated by the vision to digitize all steps and processing chemistry data, and the goal of generating and providing fair data. And before I continue with the latest achievement in the digital world, I would love to share some photos of a recent discovery in the archives of TIB. So a few days ago, a colleague found this amazing card collection of 1,000 cards of trivial names for chemicals published by the German Chemical Society in 1979. And this is just a supplement delivery for the original collection, which has 8,000 cards, but it's unfortunately missing. So I guess today you simply use an app on your smartphone and swipe through such a collection, which is updated daily, and the data is linked with many other data resources. So coming back to chemicals and our time, our focus is on data and characterizing chemicals. we are talking about the data you generate and require to characterize, describe, and identify chemical compounds. And we do this in a broader context. We look at the chemicals in reactions, their role in experiments, the analytical data like NMR, mass spectra, infrared, and many more measured for a chemical. And of course, data about the chemical or the biological activity as well. Overall, we look at a very large amount of data and data space representing information and knowledge we have about a chemical. And we aim not only to describe the data in a human readable form, but also in a machine readable form by applying controlled vocabularies, terminologies, and ontologies to semantically annotate the data. Ontologies for CHEM is one of many activities we have to digitize major steps of data generation and management on the way to FAIR data centered around the smart lab approach of NFDI for CHEM. And in Ontologies for CHEM, we develop services, tools for the semantification of data, linking data in a machine readable way. And we also provide access to ontologies and also create and develop ontologies to enable and enhance semantification in the area of chemistry data. Maybe let's take a moment to pause and examine what ontologies are more closely, and looking at the definition as defined by Gruber. So an ontology is a formal explicit specification of a shared conceptualization. And formal means here machine readable description using a formal language. Explicit specification means that concepts, properties, relation, constraints, and axioms are explicitly defined. Shared refers to agreed upon by a group or applications, and conceptualization is what defines an abstract model describing a particular field of knowledge. And in the context of machine readable data, ontologies provide a shared vocabulary that helps us to standardize the meaning of data. And therefore, they can help us to make data more interoperable by providing a common language for communication between different systems and applications and even disciplines. And this, of course, then can facilitate data integration and exchange, which is critical in many applications. You can see here that already quite a few ontologies of importance for chemistry exist, which cover a broad scope of entities. So we just heard about KBI, an ontology for chemical entities, atoms, substance groups, that also contains the few of the biological, chemical, and application roles. Less expressive and a kind of a hybrid between an application ontology and a domain ontology is a CHEM ontotaxonomy that can be used for the classification of chemicals, compounds, and which has been partially mapped to KY. Within the chemical methods ontology, CHMO, the chemical information ontology, ChemInf, or the quite new process chemistry ontology, PROCO, we can find the terms needed to describe characteristics of molecules, compounds, or mixtures. And the MOP ontology, for example, defines fundamental chemical processes like oxidation or the breaking of a covalent bond. And with the named reaction ontology, X and O, we have a pretty large collection of terms defining synthetic reactions and reaction steps. Of course, these ontologies do not cover everything we need yet, and there are some issues that need to be resolved in the future, but it's a very good start. So how does semantification of data look like? Very simplified. Looking at the description of an experimental procedure to synthesize, here in this case, 1,2,3,6 tetrahydrophthalic anhydride, we can apply many terms from the ontologies to semantically annotate the text. We do have Rx and O to defining the Euler cyclization, for example, but also defining reactions or the concept of to produce. We have the units of measurement, ontology UO, for metrical units, and the ontology for biomedical investigation provides then additional terms for and concepts for investigations, protocols, and instrumentation, to name only a few. And maybe some of you have seen a similar image before years back. So starting around, I think it was 2004, the Oscar 3 tool addressed the parsing, text mining, and automatic recognition of chemical entities, chemical names, ontology terms, and chemical data to enrich published research data and to generate machine interpretable data, but after the publication. Today, the digitization of workflow has been increased substantially, and we have the opportunity to address the creation of machine readable data early in the process of data generation, so early in the data lifecycle. So how do we manage to cementify data from the very beginning, especially in NFDI for Chem? So we provide chemistry ontologies through the NFDI for Chem terminology service, both for human and machine use, for machines and for various services. And the terms from the ontologies are used in the electronic lab notebook Chemotion, for example, as our reference implementation to annotate data. We are drop-down lists to assign methods from CHMO or reaction names from RxNO. And semantic data annotation is also provided during the upload process in the various data repositories of the NFDI for Chem Federation. And as a result, the first repositories can be queried by our overarching search service, which harvests the metadata based on controlled vocabularies from various data repositories. We can link, and better in the future, data about chemicals from different repositories, thus providing an overview of available measurement data or reaction data on a chemical. And the service will in the future furthermore act as a data hub to other infrastructures or other disciplines and their services. So to summarize, at the end, we bring all the pieces together to create something like a chemical knowledge graph with fair machine interpretable data about chemicals, and your knowledge base, they are embedded. And we are not fully there yet, but we are on the way. And there's a slide. Thank you very much for your attention. I'm looking forward for the panel discussion.
Okay, thank you very much, Oliver. And thank you for sort of giving that sort of overview of where the journey so far and, the journey still to take. And I think also that end destination that we're searching for, the smart lab. And I think Adnan talked about, tedious processes. And I think the hope is that by better classifying all our data artifacts, we could take the tedium out of things that otherwise take up our time. So thank you. Right. Reminder, questions, chat, Q&A, if you have them, whether they be specific or broad. Think about broad questions you might want to sort of ask the panel when it comes to the broader discussion. Our next speaker is someone we welcome back to this series. He took part in the first one of these, where he talked to us in sort of general terms about the data challenges associated with the development of new materials. Ken Crolin has a perspective shaped by almost a decade at NIST, and he's now helping to enable innovation in the development of better materials and chemicals. And today he's going to be talking to us about schemas for complex substances. So Ken, over to you.
Thank you very much, Ian. Now, I'm going to be leaning back a little bit more towards a historical perspective. And those of you who know me know that I tend to lean very hard into the ideas of software engineering and the practical problems that people experience when they're trying to actually work with these systems, as opposed to sort of the more high-level things. It's not so much a question of finding tools out there, but understanding what to do when you're implementing in detail. And so I'm going to be using as a case study a piece of work that I was involved with a while back at NIST, the generation of the substances subschema for the source database. The purpose of this particular database is it backs up NIST Standard Reference Database 103 A and B. And it's essentially a thermophysical property database that happens to have a substances schema so that you can identify the particular materials of interest and sort of where that information came in from the literature and make sure you're getting the right kind of predictions out the other side. So I'll only be focusing on that upper substances. Obviously, I'm going to leave the DOI for the paper up for people who are curious, so you can be a little bit slow in writing that down. And obviously reach out to me or Andrei Kazakov or Chris Musny, who are still at NIST, who'd be happy to talk about some very detailed aspects of implementing this work. At the core of this is a very, very simple table, which is the substances table, which is literally just a bunch of IDs. And the reason that it's just a bunch of IDs, it's literally just a primary key table, is because just about every identifier, when you get into the complexity of we want to talk about every single possible substance you can find in the literature, becomes ill-defined. Everyone, I'm sure everyone on this call is familiar with the problems with names. They become very useful for looking things up, and they're very good as a display feature when it comes to talking about identifying a certain material, but they're oftentimes ill-defined, and really there's just so many of them, you don't really know what to do. Registry numbers, a classic problem with, let's say, the CAS registry number, is that you have duplicate numbers for the same material, and you oftentimes have to deal with deprecating numbers. But if you're pulling information from the literature, you need to be able to figure out what they would have referenced it with back in 1970, which wouldn't necessarily be the same number they're going to reference today. As well, there's also a time unsync problem where oftentimes, this is a little better today than when this was first put together, you haven't actually assigned a CAS number to a material when it's a novel, when it's material that's just been discovered, a novel ionic liquid that people are proposing for some sort of a complex separation process. And so you end up with a situation where you can't satisfy from a rigorous perspective, from the computer's perspective, you can't satisfy all the fields that one might want to put into this database. And that's really the heart of what of the challenges I want to try and discuss today, is that you have this situation where there's an unsync between the requirements of the computer, the knowledge that people have, and the fact that as you sort of get into some moderately complex situation, you sort of have an explosion, proliferation of additional information needed, sort of nested complexity. And I'm also, you'll have to excuse me, I chose this scheme in particular because it gets into chemical complexes, and I just couldn't resist the pun. And then the last bit that we have an issue here is you have images. This is a necessary display feature. People need to be able to see the molecules. Layout engines oftentimes aren't as good as we might like, and so you really do need, again, the kind of manual curation, subtle interaction. This can't be a single insert into a database. It's sort of a slow evolution of analysis. We have a lot of the kinds of tools that Adnan was kind enough to sort of give us screenshots of in talking about Kimby. And so it's just sort of like, how does all this stuff get put together? Now we get to the actual definitions of the compounds. The most basic thing is a compound is anything that's going to have a single well-defined INCHI, or well-defined chemical structure, something we can convert into an INCHI, something that we can create a INCHI key off of and use that INCHI key to detect collisions within materials. And then that gives us four things that have this property and ability to build a table off of this, have a secondary table with some larger elements to it so we don't actually interfere with the database speed, because this is a very practical sort of thing. But it's actually interesting because even at this level, we're already having problems with INCHI as defining our chemical compounds. Because, for example, INCHI by default doesn't really deal with tautomers very well. And so you can play with flags, you can add some extra features to make sure that that's not going to collide. But the default flag, for example, on the INCHI key generator won't actually give you something that performs adequately there when you do need to talk in specificity about the tautomers. And then you also have the flip side of oftentimes your consumer doesn't actually care about the tautomers, and they actually want to combine all the thermophysical properties for all these measurements. And so you need that kind of flexibility built in here. You also have the problem of things, we also need to disambiguate things like, are you talking about a monomer or are you talking about a dimer? Where if you're measuring something like the vapor pressure of formaldehyde, association in the gas phase is so substantial that you can't actually measure things that people think that they're actually measuring. And so you get a situation where something's mostly okay, but you have to start adding these little bits of complexity to satisfy the computer's need for uniqueness. And at the same time, get into a little bit more, have it general enough that when the chemist comes to the platform, they understand and their answers are actually answered the way they intend them to be. we have in contrast to compound, we have something that we can't, additional things we can't define this for. For example, allotropes. You need to be able to specify graphite because if you're talking about chemistry, if you're talking about enthalpies of formation, enthalpies of combustion, those are two different carbon forms you're going to have to reference in terms of those reactions. But the inchi for monatomic carbon and the inchi for graphite are actually identical. And then there's also other things like diatomaceous earth that you want to reference. And then lastly, we get to the crux of the whole thing, which is the complex table. And for this complex table, we're trying to reference things with disconnected structures, but are still well defined. Hydrates, for example, or if you're talking about ionic compounds. One of the big problems we had to deal with when we were designing this was mixtures of ionic liquids, where you have 4 different ions in fixed compositions, but the way that a experimentalist is going to report that information is in terms of raw ions that they poured in there, but you don't want to lose the fact that the permutation there on those pairings is actually another perfectly lovely way of expressing that chemistry. And so you create a component table that links these elements together. And so you end up essentially with a situation where the same material has the one entry in the substances heading, but has entries in both the complex and compound schema. So you're actually going through this information in multiple paths. So to close up, and I'm a little bit over, I apologize. The heart of what I'm trying to communicate here is you really have to meet complexity where you meet it. You have to sort of, if we didn't have very specific use cases, it would be a terrible idea to have introduced any of these complexities. So when you're working with software engineers, when you're working with kind of trying to get these things into code, you really have to think about the question, what is the problem I'm trying to solve? Am I actually giving an efficient pathway to get to a solution to that problem? And am I satisfying exactly that need? And in this case, we've actually expressed complex concepts in about 3 different ways for the idea of, things that don't have very clearly defined chemical structures, but each one matches the actual application. So thank you very much, and I look forward to the panel discussion following.
Thank you, Ken. That was super sort of like elaboration on the realities that are out there when it comes to dealing with chemistry data in the real world. And I think some interesting observations on the interface between machines and humans. And I was sitting there thinking, well, if humans were out of the equation, would it be so much simpler? But I think you heard at the beginning, there's still that role for humans to be able to curate what comes out of the machines. So ChatGPT ain't done away with us yet. And there's still a lot of road to travel and some challenges for this wider community, I think. So to finish off with the talks now, we're going to continue on this sort of theme here of more complex sort of like mixtures of molecules, and we're going to hear from Alex Clark, who spent many years working in areas of computational drug discovery, currently at Collaborative Drug Discovery. If you dig into his past, you'll find he started in the area of metal organic chemistry, and he's brought his broad and diverse background to bear on a number of innovations in cheminformatics, and looking forward to hearing what he's got to say about the work he's been involved in mixtures. So Alex, over to you.
All right, chemical mixtures. So the basic premise here in a nutshell is that the cheminformatics industry is based on our ability to represent molecules as these graphs of atoms and bonds. And this has been incredibly successful because it really is quite a good correlation between your data structure and what's going on with chemical reality at the atomic level. And it's also very convenient to go back and forth between these graph representations and what chemists actually want to see as diagrams, and also graphs are very convenient for computers to work with. So we've got decades of success stories in modeling what molecules can actually do. However, the very idea that a molecule is the actual thing that you're handling in the real world is itself an approximation that's not always very valid. So for instance, this structure here, it's the active ingredient of aspirin. And if I were to show you that structure, you might think I'm talking about the pure compound, or you might be thinking of the formulated drug pill that you have in your bathroom. Or perhaps it's something else, like some amount of this dissolved up in a mixed solvent. And so these 3 different concepts are really wildly different things. They don't have quite as much in common as you might think. So recording this context information for mixed chemicals is incredibly important for most use cases. So just by way of example, as a chemist, the first thing I think of is fine chemicals, and these are mostly well-defined chemical substances that you can buy, and they're very rarely 100% pure, and many times there are deliberately added adjuncts or solvents or so on. And so having the information about what is actually in the bottle is generally quite important for what you're doing. On the other hand, there's drug formulations, and these are usually made up of 1 or 2 active ingredients that do the actual biology, but there are also a whole lot of excipients mixed in as well. And so if you vary the concentration of the active ingredients or you change the excipients up, you've got a different drug, and it is incredibly important to capture that information in a way that computers can use it. Elsewhere in the realm of consumer products, vast industry, obviously, but most of these have some number of substances in them that can be well described using chemical notation, and tracking that is incredibly important. In the environmental health and safety realm, the difference between the safety profile of a pure compound or that same compound mixed with other substances is often quite radically different. So it's really essential to make sure that you're reading the right material safety data sheet that applies to the substance that you're actually working with. Otherwise, the results could be quite catastrophic. And similarly, with regulated substances, there are hundreds of thousands of materials that have laws passed about them. And a great many of these are mixtures, and a lot of them are very hard to actually describe. And so many times it's very difficult to tell if you even have one of these things. And that's kind of important. If there's a law about it, you need to know what it is and when it actually applies. And that's a very challenging area of mixtures. So the basic idea of machine readable mixtures is really comes down to this idea of a hierarchy. So a good example to illustrate this is something you can buy. It's a common solution of a well-used organic reagent called lithium diisopropylamide. And so the first split point here is we can divide it up into 2 components. The first component is the active ingredient. That's the LDA itself, which we can represent using atoms and bonds. That's nice and clean. It's a well-defined chemical component. And it has also very precisely measured concentration. So that's the part that you're really interested in. But the other part that makes up the substance is what I'm just calling solvent, and that's essentially a placeholder. And so there's no concentration information there because it's just, which means it's presumed to make up the remainder of the space in the vessel. And so the solvent itself is defined as a 1:7 ratio of THF and hexanes. Now, THF is a well-defined molecule, so we can draw it as a structure. But hexanes, on the other hand, hexanes plural, is not a chemical. It is, in fact, an ensemble of a variety of other hydrocarbons, and it tends to be rather poorly defined because you don't care. It's just something that's been distilled over a certain fraction, and it's cheap because it hasn't been well purified. And if that doesn't matter, then that really is the right thing to do because you save money. Now, the important thing to note about this hierarchy is it's not only can it capture this idea of mixtures, so each node in this tree is, if you like, something that could be in a bottle all by itself. But there's also the ability to localize the uncertainties and errors. So as you cascade down this tree, the amount of vagueness about what you have increases. And so on the top left there, you have a very precise measurement of the active ingredient because that matters. But when you start to dig into what's actually in this bottle of hexanes, it gets less and less specific. And that's okay because it's the reality, and that describes as much information as you need. So the last thing I have to say about the hierarchy is you can also attach metadata to any point in this tree, and that has nuance because, for example, if we have physical properties of that are attached to THF, we're referring to a pure compound. But if we attach properties to this thing called hexanes, we're actually talking about the mixture within the mixture. And so where you associate this extra metadata in this hierarchical structure has a lot of really important meaning. And very often that information is lost in many of the representations that people use currently. So describing this as an actual chemical data structure, at CDD, we use a format that we call mix file. So it's relatively verbose. It's kind of like the mixtures version of a mol file. So you capture information about the structures that make it nice and easy to make pleasant diagrams to people. But the motivation for this project originally was to provide an upstream data source for the proposed mixtures NG notation. And in a very short nutshell, this involves taking all of the structures you have access to in the mixture, creating an NG for each one, taking the sorted unique list and basically gluing them together for the beginning of the Minci notation, and then following that up with information about the hierarchy and the concentration. Now, this has a couple of very useful properties that are inherited from the INCI identifier, and that it's very easy to index the individual substances. You can just do some trivial string manipulation on the database. You don't need a fancy cheminformatics toolkit or anything. So that automates out a lot of otherwise difficult tasks. And also you can compare two Mincis to see if they have the exact same list of substances just by looking at the beginning part with the part in blue. So you can quickly do a preliminary, are these the same thing? And then follow up by analyzing the concentrations, which get a little bit more mixed, if you like. There are many different use cases that you have to consider when it comes to building software, and the important one that you should start with is how do you get your mixtures information in this machine readable format. And there are several options. One is to use the open source mixture diagram editor that we put out there. If you're a user of CDD Vault, we have functionality for upgrading incoming Excel spreadsheets, which a lot of people use, and we can basically form the columns into mixture components. Or you can look them up in databases or use our machine learning interpretation for name descriptions. So yeah, multiple ways of getting content in, but once the content is in there, that's when the fun really begins. Because once the machine readable mixtures formats make it very easy to index the individual components, whether that's by structure or name, metadata properties and so on, so we can very quickly see what is in each one and look them up. And we can also imagine all kinds of more sophisticated queries that we can compose, like for example, does it have a certain structure and is it within a certain concentration range? Or we can look for metadata identifiers and other properties, or we could filter out and remove certain substructures. So you can imagine if you're scanning through hundreds of thousands or millions of mixture entries, looking for mixtures with certain properties, you can compose fairly elaborate queries and zoom in on the ones that you're actually looking for. And last but not least, once we have large amounts of information in this machine readable form that's associated with actual measured properties, like things like solubility, for example, but pretty much anything else, you can come up with all kinds of creative new ways to build models using this information. This is something that it's in a very nascent form right now, but it's really going to keep us busy for some number of decades, just like the original structure activity modeling. So that's mixtures in a nutshell. Just some final comments if you want to read any more about this. There are a couple of GitHub sites that you should definitely take a look at. We originally published this mixture information in Journal of Cheminformatics in 2019. It covers a lot of the content. And also more recently, we contributed a section in an Environmental Science and Technology Review, which is talking about UVCBs, which are the really hard to characterize mixtures in regulatory contexts. So that's really state of the art. So that brings me to an end, and I'll be happy to answer any questions in the follow-up panel.
Brilliant. Thank you very much, Alex. And I think that has pretty much landed me to where I was going to actually start the panel discussion. And so I'm going to ask you a question first. Then we'll be tracking back through the speakers to the beginning of this particular case. So just to give you fair warning, but please, everybody who's here participating, please don't be shy in indicating if you yourself want to ask a question, whether it's specific or general, either in the chat or stick your hand up in Zoom, one or the other. But I think you started by saying, with observing that, it's 40 odd years ago, right, that we were sort of started looking at how to reliably represent, discrete organic molecules. And obviously a lot of road traveled. And then you said at the end, there's a lot of road to travel now with the mixtures. So I suppose what I want to tease out in some of this panel is a discussion is what are the areas we should be focusing our innovation on at the moment that are going to give us most impact now in order to sort of advance things so that our chemical data infrastructure can more realistically capture what's going on in the real world.
So you mean to ask the domain that we should be modeling, or how to capture the data?
I think if there are domain, I think either if there are specific domains that it would be worth focusing on, but I think also what are the sort of technical challenges or social cultural challenges just generally that we need to be focusing our efforts on, so we get to a point where what we can do with discrete molecules in terms of the vast amount of machine learning and modeling and data management we can do for these more complex mixtures and components and stuff.
I'll do a quick answer and let other people chime in. So for domain, I think the obvious low-hanging fruit is probably solubility, because it's just such a natural way to describe two things together and how much of this goes into that. And the value proposition of having better solubility models is huge. People are screaming for solutions to this in real-world situations. In terms of capturing more data, obviously, I'd prefer it if people use the formats that we like most, like the mix file and Minchi, but most mixtures in the chemical realm, at least, are really quite simple. I mean, you have two things and one of them has a percentage and you can work the rest out. And as long as you can record that in a form that can definitely be tracked to a structure. I mean, if it's, if you have a CAS number or a name that is unambiguous or an Minchi or anything, if you can put those in, you can put those in 3 columns in a table. And if you just make sure that those can be transformed to a standard structure like Minchi or mix file, then we can just do that with a simple script and accumulate it in a database. So from a getting started point of view, that is maybe the way to think about it. But certainly building scripts that can go through sort of slightly less tractable data sets and reliably put together a well-defined mixture is a valuable exercise.
Thank you. Kind of like meeting people where they are now, not trying to get them to change their way of doing things. So Ken, your observations on this particular area?
Was that a prompt? Yeah.
Yes, it was a prompt. Sorry.
One thing I actually loved about Alex's presentation, and I don't know if people noticed it, was that he pointed out that the hexane solvent component of his solvent had a boiling point range. And that's one of those nasty things that people don't realize when they're putting together these chemical schemas, where all of a sudden something that you thought was going to be a well-defined concept becomes ambiguous or gains a distribution. And so again, it's these surprising things just sort of pop up at you as you're actually engaging with real world data, not the clean things that we want. You know, ideally, you'd like to think of, oh, it's just an ethanol water solution that we're putting some aspirin into. But even that, impurities and other things sort of get complex. And it can be very, very hard, especially for people who have been trained in chemistry, to focus on nitty-gritty details, to keep in mind what actually matters for the problem they're solving and whether or not it's going to be relevant versus things that are either avoidable today or possibly just never going to be relevant to what you're trying to accomplish. And that also is, very, very important when you get into FAIR concepts around interoperability, because it's those gaps in application that often inform the ways in which the schemas between two different data sources have a lot of difficulty reconciling with each other.
Thank you. Oliver, how can ontologies help? Where are the areas where we need to develop the ontologies to help?
Yeah, we need to apply them, right? So I was fascinated by these complex use cases because that's reality, right? And so we start modeling with the easy cases to build up the software systems and to apply the first easy ontologies and start to getting more and more complex. And I do see a huge potential in this FAIR data approach and having more and more data available on the one side, but also always have to keep in mind the data quality. So how can we validate data to make sure that, especially the published data, is actually what's inside what it's described, right? So having or providing schemas during the upload process or the publication process to have hopefully mostly automatic validation of data and metadata. That will be one of the big challenges.
Okay, thank you. And Adnan, in terms of your challenges creating KEBI and curating it, whereas the complexity, where was the challenges that would make a big difference to your tedious processes as you described them?
Yeah, so KEBI is manually created. So I mean, the ontology terms, we have to manually add each chemical into those classes. I think having some sort of automated tool which can do that, I think that would be a significant advantage. I think with chemical classification, a lot of groups are doing things their own ways. I think having IUPAC's input in this would help, especially developing a standard ontology for chemical classification would really help. For example, IUPAC can propose better classifications or better terms for those classifications.
Okay, that's an interesting comment, actually. And then when you say chemical classification there, you are talking about that sort of, it's a furan, it's thinking about that from a sort of, describing the chemical. Okay, yeah, I mean, I'm not speaking for IUPAC here. I guess I've always looked at what's going on in the wider community and think actually people are doing this, but I think what you're kind of maybe suggesting is that.
But I think having for example, the chemistry community consensus on this would help,
Yeah, absolutely. So, yeah. That's a very good point to make. Henry, your experiences, if you want to bring this to MIME types, that's great, but I'm also thinking of your experiences putting together repositories for chemical data, and maybe something there that speaks to the complexities that we're talking about here.
Complexity is the right word. I mean, I'm interested in, everybody's discussed these sort of multiple layers that any system can have, the complexity of hexanes, etc. Media types obviously are just a two-layer model, a primary one and a secondary one, and they don't really lend themselves to third and fourth and fifth layers. So that's a very sort of very crude finding tool, if you like. And then having found the data, you then have to decide what extra layers you're interested in. If I may seek your indulgence, I'm putting into the chat a link, which I think illustrates nicely these layers, and some comments I think that Alex made, which is that you've got the standard representation of a molecule, a connection table with bonds or not. But as a computational chemist, I'm discovering deeper and deeper layers below these simple representations. If you wish, go to the DOI that I've just offered you and click on the hyperlink called isosurface. And it's a bit slow because it's actually quite a large isosurface, and it'll take maybe on your computer a few seconds to load. And you'll see, if you've done it, all sorts of features colored in cyan, green, yellow, a few red ones, orange, etc. These are all layers over and above what we capture when we draw a bond between two atoms. And I have no idea, in terms of sort of layered models that we've been talking about today, how you start capturing these subtleties. Every one of those features is important for enzyme recognition, catalysis, you name it. Quite often just one of those features can account for the stereoselectivity, let's say, of a chemical reaction. So you can't ignore them, but how do you capture them in layers? So I think other people have made the very valid point that we're just on the beginning of a journey. It's been a long journey, and it's a way to go. And every time we think it's coming to an end, we'll find another depth and a few more layers that we need to know about. I think we've got to have infrastructures that are able to cope in the future with these unexpected layers that we don't even know exist at the moment in molecules, and which the standard chemoinformatics tools are not yet really capable of expressing. I'm not sure I answered your question, Ian, but I thought those are the thoughts that came into my mind when listening to the other talks anyway.
No, I think you provide good food for thought, and you know, I think I can track this back to some of the work we do at CCDC, where we're wanting to map features into protein active sites, and now people are saying, well, how can I interoperate with these as a machine? And then it's a question, well, how do you take these visualizations and capture them in a way that a machine can understand? And I
Yes, it'd be
think
great, for example, to have a machine go through all your 1.2 million structures and compute these extra layers automatically. But again, I think we're a long way from doing that, and not least of which, many of these additional computations can take days, if not weeks, of computing time to do. So,
was going
where
to be
do
my
you do
question.
things in real time, and where do you send it off to a batch process to grind away for days to get the result?
But that then adds to the sort of value actually is if you do compute these things of storing them in the ways that are readily reusable and interoperable, because you really don't want to have to repeat that. And it's that kind of, yeah, what's worth. And I think that coming back to the hexades example, I love the sort of comment that Alex, you made there about, you know, we don't care too much about what those proportions are because it's all about what's cheap at that particular time. I think there were themes coming through, and I think it started a little bit there with Adnan as well, talking about we kind of like, you know, wanting community agreement on the classifications. And I'm wondering where's the role for standardization here that can help us maybe conceptually as much as in terms of standard descriptions of specific entities. I'm going to jump into Oliver here because I think you kind of like this might be something, you know, that you've got a perspective on. Is, you know, there's a risk that lots of different initiatives are going to come up with different solutions that somehow are incompatible, or there's a risk that they do that because every different sort of circumstance is different. But do you have a feel for where there could be value in community consensus on what our standard approaches to dealing with this complexity?
Yeah, maybe I pick up the comment about IUPAC and what an IUPAC can do on standardization, and just as a comment that we, with our work on ontologies for chemistry, and discussing how, for example, the IUPAC Gold Book can be used for the definition and description of ontology terms, so that we have a reference, a ground source of truth. That's one thing. And maybe concerning this interoperability and especially alignment for the world of ontologies, we are trying to bring together the ontology community in chemistry, or working with chemistry ontologies, with our ontologies for chem workshops we do once a year. And also we are collaborating and discussing across the different scientific disciplines within the NFDI, especially the natural sciences and life sciences, bringing together to at least try on that level, bring up some kind of, I'm not speaking about standardization, but discussing what we do and pick up ideas others have and try at least to align a little bit.
Mm-hmm. And I think, you know, if we track back to sort of like the very first webinar of this series, we were keen to bring in voices from other disciplines, and I think that's an important part of this. Of course, chemistry is the central science. It's special, it's different from all the others, but some of these challenges are going to be very, very common. Ken, any thoughts from you on, you know, standardization at a conceptual level as much as a sort of entity level?
I mean, I've always been a fan of community standards and community evolution, but those almost always follow from an awful lot of prototypes and an awful lot of experimentation. I do think that we've had, enough time to work on a lot of these problems, that we've had some fantastic standards come out and some fantastic solutions. But ultimately, I think this is just going to be a situation where where you have a marketplace of competing ideas, and the thing that actually solves the problems that people actually have will get the funding and will grow. I think that if you want to be the person behind that solution, you should probably keep an eye towards making your tools easy to use, and you should keep an eye towards where the real pain points that people are encountering are. The number of times I've seen people sort of want to develop a standard or want to solve something that is a non-problem from like an industrial perspective is not small. Like where you see something that there's a philosophical inconsistency, but the act of changing the technology over isn't going to actually impact people's day-to-day practice in a meaningful way. That's not an avenue to pursue if you want to have great impact.
Okay, and that sort of reflects, I think, what you said, the importance of having this use cases that sort of drive this, and make sure that what we're doing has relevance.
Yeah, and I see a question directed at me in the chat. I mean, the answer on tautomers is actually very similar to the answer around the hexanes mixture question. Does this different from a data storage perspective? You have to make sure that you're actually correctly capturing in a machine actionable way the information that was represented. But in the consumption of that data, you need to have sort of flexible consumption pathways so that something like a tautomer will end up in front of the user that needs to use it if the question they're asking is insensitive to that distinction. And then in this case, you can have an INCHI for each individual conformer, tautomer, whatever you call those sub-elements of a tautomer, tautomeric structure. But you really want to make sure that the logic there aligns with the physics and the need. Hopefully that answers your question, Jonathan.
Thank you. Yes, very, very helpful.
Thank you. Alex, any comments from you either on the sort of general issue of sort of like standards or areas where IUPAC could be thinking of going next, or if you want to talk about tautomers?
Maybe I'll just comment about something slightly different that should relate to all of the subjects, is that we really have discussed and thought quite a bit about the idea of building an ecosystem of these technologies and tools and mixtures and everything else that we're doing in this group. I think of it as like building a tower out of bricks. If you just stack one side up again on top of each other, it's not going to work out. You have to build a series of protocols, get some data out there, have open source tools that everyone can use, but also have commercial tools that cost money but make people's life more convenient. You have to have outreach. You have to meet users where they are, but also pay attention to the philosophical abstract details of doing it right. You got to have all of these pieces, but you just build one brick at a time, and that's kind of how we're thinking about that. So sorry for the answer to the wrong question, but I thought that was worth mentioning.
Well, once again, you've magically sort of led me on to another area I wanted to sort of explore finally, I think, as a final round of questions here, and that is who else we should be working with, bringing to the table to involve in these discussions to maximize chances of success or bring in other experiences. And you mentioned, for example, software vendors, and they play an important role because they got to build systems, and that's what people are going to use. In addition to software vendors, any other sort of parties that you think are missing or we could do well to involve more?
I'll just throw in one and then people can follow up, but just identifying people out there in the world of real industrial problems that have a very specific problem that might not be the one you're trying to solve exactly. So maybe it's elaborating on what Ken was saying, but I can think of one that I didn't anticipate at all, is useful drug antibody conjugates. Apparently the mixtures data structure is very helpful for that. It's not designed for it at all, but these are people who have a specific problem, there's no solution, and our tools just happen to work better than anything else. And so they're pushing it. And but people like that can drive a big part of a whole ecosystem, because it really is solving a real problem, and there's money involved and interest, people put in time. And so just as many examples like that as possible. And I suppose another one would be the UVCBs, like the regulatory examples. That's a huge problem, and there just aren't any answers to it. And if we can provide actual practical solutions to even some of it, that gets people's attention. So stuff like that.
Thank you. Adnan, from your perspective, coming from a more structural biology or just biological area, are there people we should be involving in these discussions?
I think we should be involving the chemists, the our major users are the end users who are actually using the database. So we can involve them, and the ontology community is probably one of the users that we can actually get involved.
Okay, yeah. There's experience there from people who generally understand how to conceptualize things. Oliver, do you want to maybe elaborate on whether it's ontologies or other parties that we should be bringing in?
Yeah, parties. So there's one big question mark, right? So thinking about getting more and more data out there and thinking about machine learning and in future applications. So how do we know what data to archive or publish and how to describe it when we don't know all the future applications and use cases? So what's to come there with machine learning?
Interesting question.
Well, specifically, again, as a computational chemist, and I actually put a comment in the chat. For years, I've been mortified by the fact that most repositories out there, the Zenodos and the other ones, that there are very few examples of repositories which capture rich data, such as wave functions. Nowadays, most people compute a wave function for a molecule of interest. We won't argue about which model they use, etc. But almost never do they then share that wave function with anybody else. Okay, maybe to interoperate, you might want to have a slightly different wave function that's better at doing something than the one that it was computed for. But recalculating a wave function in a slightly different way, different basis set, etc., is actually relatively trivial compared to starting from scratch. So I echo this idea that we should be engaging more and more people to provide rich data. Throw away as little as possible. In computational chemistry, that's the wave function. In NMR spectroscopy, that's the raw free induction decay data that comes out of an instrument, and the same would go for pretty much any instrument. Most people process instrumental data and then share the processed data, but why not share the unprocessed data, the loss-free data? So that's the main message that I want to sort of put in people's minds, is that we're too often throwing away data unnecessarily.
Okay, thank you. And following on from that, Ken, I think you get the last word on the panel discussion.
Yeah, I mean, I would say that a group that is absolutely essential here is the chemical engineers. It's the process simulator developers. It's because it really is, chemistry in a lab is neat and you can develop a lot of great molecules, but it really is putting the manufacture into practice of practical systems, whether it be drug systems, whether it be fuel systems, whether it be just consumer products. It really is making sure that they have the data that they need and they have the systems and interoperability and the ability to collapse complexity where they need to be able to do so in order to actually develop a system and have one number to plug into a differential equation to figure out how to size a heat transfer system, the heat exchanger. It really is just a question of, can we get the right information to the right people, and the consumers, and really the chemical engineers and the process simulators in my mind, but sure, there's other folks that are doing that. Using this for design and using this for engineering are essential. Okay,
thank you very much. That's a great point to end on. So I will just conclude my part by thanking again for the speakers for their presentations, but also a very informative and thoughtful discussion. Very much appreciated. And I will now hand back to Fatima for some closing words. Thank you very much.
Thank you, Ian, for moderating this session, and thank you everyone for sharing your expertise. I'll share my screen now, and I think what we're gonna have right now is we would love to hear your thoughts about this webinar and how we could help and serve our community. Please let us know. I will launch a poll right now. We have a couple of questions, so it will be great if you answer those. There are like 5 questions. Meanwhile, I'll be happy as well and glad to share with you our future events, actually, we have a very busy month of March. So we'll be hosted by Convoices webinar. We will deliver kind of conversation as well about what is digital IUPACs and what is our project also is doing. We will be also showcasing some of the early prototypes as well of our project in that webinar. We will be also present in the RDA sessions in multiple in-person and in virtual ones. So stay tuned, and if you are there as well, catch up with our team also in RDA, and that will be in March 20th to March 23rd. Also, we will be present in the ACS spring meeting. So if you are there, if you can make it, it's a hybrid actually meeting. So we will be in Indianapolis in March 27. We'll have a one-day workshop. The morning session will be hybrid. So we'll be also showcasing our prototypes and our deliverables of the project. In the evening session, it will be more hands-on and it will be a workathon. So to test those prototypes and having the community feedback. So if you are there also, please let us know and join us in the ACS spring meeting in Indianapolis. Also, those webinars, actually, this webinar is part of a series of 4 webinars. 3 of them are already uploaded in the IUPAC YouTube channel. So if you want to rewatch them, they are also there. This webinar also is recorded and it will be uploaded very soon to the playlist of the IUPAC. I invite you all to connect with us through our social media. We have a Twitter, through our email, and through the IUPAC channels, the website, and the Facebook as well. With that, I'll come to acknowledge all of you and thank you everyone who attended, and for our speakers who also graciously accepted to be with us here and share their experiences. And I wish you all a great weekend. Bye-bye.